Dear students, welcome to Tungal online classes. Today we'll start with the biological significance of carbohydrates. Right? So yesterday we had gone through what are carbohydrates, what are the types of carbohydrates, that is monosaccharides, oligosaccharides as well as polysaccharides, and how polysaccharides are more useful. So how monosaccharides as well as oligosaccharides are more useful. So simply we have gone through that one, and totally now today the biological importance or the significance of these carbohydrates totally we are going to learn over here now. Right? Let us start with the biological significance of carbohydrates. Biological significance of carbohydrates. Carbohydrates have their own significance in plants as well as animals. How they are very important in plants as well as animals, that we are going to learn now. Right? As you know, the total dry weight of the plants, so that is about 60 to 90% of the plant's body itself consisting of these carbohydrates. Right? Carbohydrates makes the 60 to 90% of the dry weight of the plants. Right? This is one significance of what? Carbohydrates. Carbohydrates makes 60 to 90% of total dry weight in plants. That's why this is more important aspect to be remembered. Okay, and so these polysaccharides especially, polysaccharides as well as even monosaccharides, oligosaccharides, they are acting as the storage products. Right? So see here, starch is one of the polysaccharide which is a stored food material. Stored food in case of plants. Right? That's why storage material it is acting as. In the same way, glycogen is also another carbohydrate which is the stored food for animals. So that's why some of themselves are acting as a stored food materials. And next one, some of the oligosaccharides, as we know better that oligosaccharides chains are present on the outer surface of the cell membrane. They are acting as cell surface recognition molecules or cell surface recognition structures. Right? So that's why oligosaccharides act as cell surface recognition centers. They are acting as. Right? This is also more important one. And one more is heparin. Heparin itself acts as an anticoagulant in blood itself. Right? So that's why heparin, it acts as anticoagulant in blood. So this is also one more significance of what? Significance of these polysaccharides. Even polysaccharides like cellulose. Fifth, cellulose and chitin. So they are acting as what? So see here, cellulose acts as the cell wall material in plants. Cell wall material in plant cell. It is acting as cell wall material in plant cell, And chitin acts as what? Exoskeleton. Exoskeleton of arthropods. One thing. And even chitin acts as the cell wall material. Cell wall material in case of fungi. So that's why cellulose and chitin are biologically very important one, right? And the sixth one. What is the sixth one? See here, many of the carbohydrates like trioses, 3 carbon containing compounds. So what are these 3 carbon containing compounds? So that is, one is glyceraldehyde as well as dihydroxyacetone, right? So one is the triose like glyceraldehyde, and glyceraldehyde is one, as well as dihydroxyacetone. So these are also the trioses, means 3 carbon containing compounds. These are sugars. So these mainly play a very important role in Kelvin cycle of photosynthesis. They play important role in Kelvin cycle of one thing. And the second one, glycolysis cycle is there in respiration in plants. So these trioses play very important role over here, right? So dihydroxyacetone as well as glyceraldehyde are playing very important role in Kelvin cycle of photosynthesis. Photosynthesis occurs by Kelvin cycle itself, and one more cycle, that is glycolysis, that take place in respiration in plants. So in these two cycles, these two triose sugars play a very important role. They are the intermediate compounds they form, right? So this is also one more thing. And next, these carbohydrates play very important role. How they are playing a very important role? Some of the sugars like glucose, fructose, as well as galactose. So these sugars, they are acting as respiratory substrates. They are acting as the respiratory substrate during respiration. The breakdown of these sugars take place, that is glucose by glycolysis. Glycan means sugars, lysis means breakdown. See here, breakdown of glucose, fructose as well as galactose that take place, right? So in presence of air or oxygen, to release energy, they are acting as a respiratory substrate and help in releasing energy as a result of these cycles. So this is also one more significance of what? So these sugars itself. And so one more thing is 8-1. We can write here itself, right? Good to write. So see here, one sugar, that is pentose sugars. Pentose sugars. Like, what are those pentose sugars? So we know deoxyribose is one pentose sugar and ribose is another pentose sugar, and these two are the major constitution of DNA as well as RNA. They are the major components of DNA as well as RNA, means these sugars help in the synthesis of the genetical materials like DNA as well as RNA. So this is also one more fact to be remembered, right? And ribose is another one. Ninth one I shall write here. So ribose is also used in producing some of the energy packets like ATP, right? And so even some of the NAD coenzymes, NADP. So all these are EFMN. Okay, all these are synthesized with the help of ribose sugar itself, right? So this is also one more function of or the significance of these sugars itself, right? One thing. And hyaluronic acid is one of the hyaluronic acid. Hyaluronic acid. So this is the cementing layer or cementing materials that lies between two animal cells. Right? So means it makes the cementing layer between two adjacent cells in animals. Right? So this is also one more significance of what? These carbohydrates. Right? So I have discussed very few number of what? The biological significance of carbohydrates now, and let us start with another biomolecules, very interesting one, and that acts as, means those biological molecules, they act as the genetical materials over here. They are nothing but nucleic acids. What are these nucleic acids? So how they are biologically very important, so how they are said to be as genetical materials, who discovered them, everything that we discuss now. Yes, the next one is nucleic acids. Let us start with nucleic acids. Another biomolecules, nucleic acids. What these nucleic acids are? These nucleic acids are the macromolecules. These are the macromolecules which are present inside the nucleus and help in transfer of genetical information from one generation to another generation. They are called as nucleic acids. Right? So let us write the definition. These are the macromolecules. Macromolecules present inside the nucleus and help in transfer of genetical information from one generation to another, called as nucleic acids. Right? So these are the macromolecules I cite. So biomacromolecules, biomicromolecules. So let us differentiate that one here also. Right? So let us see. Nucleic acids, these are the macromolecules. See here, this is one animal cell and it has a large nucleus at the center, and there you come across at the center of this nucleus. So inside the nucleus there is a thread-like material, right? And that thread-like material is called as what? The chromatin network. Chromatin network it is called as. And this chromatin network contains so as to form chromosomes. Chromosomes during what? During cell division. This is the fact which is known to us, of course. And these chromosomes are made up of what? DNA. So RNA, very limited RNA, as well as proteins. So what is this DNA? This is one of the genetical material. This is one of the what? The nucleic acid, which is present inside the nucleus, which help in the transfer of genetical information from one generation to another generation. This DNA itself is present in the chromosome. Chromosomes are said to be as hereditary vehicles. Means what? They help in transfer of genetical information from one generation to another generation. Right? Indirectly, this DNA help in transfer of genetical information from one generation to another generation. That is parents to offsprings. Right? That's why. So these are nothing but nucleic acids, where they are present inside the nucleus. What do? They help in transfer of genetical information from one generation to another generation, from parents to offsprings or young ones or child, right? Parent to child, right? So like this, genetical information is transferred through these nucleic acids. Is it very clear? So what are those nucleic acids? One is DNA, another one is RNA. I come to this point later. Okay. So now we came to know what are nucleic acids. These are nothing but macromolecules. These are the large biomolecules. So these are present inside the nucleus, which help in transfer of genetical information from one generation to another generation. And so Frederick Marcher is the first name of the scientist, the one who discovered these nucleic acids for the first time inside the nucleus of pus cells. Frederick Marcher discovered nucleic acids in pus cells for the first time, and he named them as there he saw under the microscope there is a thin thread-like material inside, so the nucleus, and that is called as nuclein by himself. Nuclein, he called it as nuclein, right? So basically he called it as nuclein, means inside the nucleus, the material present inside the nucleus he called it as. And later, so Altman is another scientist. The one who named them as nucleic acids. Right? The one who named them as nucleic acids. Why? Because they are present inside the nucleus and they are acidic in nature. They are present inside the nucleus, and so they are acidic in nature. Why they are acidic in nature? Later I will go through that. So they are acidic in nature. Because of this reason only he called them as nucleic acids. Right? So one who discovered nucleic acids are Frederick Marcher, was a doctor scientist, the one who was working on some cells. Right, in the pus cells he saw a thread-like material, he called it as nuclein, and later Altman named them as nucleic acids. Right, Altman named them as nucleic acids. Right, so these are the scientist name that you must remember here. So he discovered them in the year 1868, right, and in the year 1899 he named them as nucleic acids for the first time. Okay, so now we are very clear that, so who discovered these nucleic acids, where did they discover these nucleic acids for the first time? So let us see the next thing. So there are two types of nucleic acids are there. Two types of nucleic acids are there. So one is DNA, deoxyribonucleic acid, and second one is RNA, ribonucleic acid. So already you know deoxyribose is the pentose sugar present in it, and ribose is another pentose sugar present in RNA. These are the two types of nucleic acids which are present over here. Okay, so let us go through the occurrence of these nucleic acids. Occurrence. Where do you get these nucleic acids? Where do you see these nucleic acids? They are always present inside the nucleus. Inside the nucleus. Right? So they are present inside the nucleus. Of course. So this is also forming nucleoid. Nucleoid is present in the bacterial cell, that is prokaryotic cell. Nucleoid is formed by what? This nucleic acids only. So inside the nucleus of what? All eukaryotes. Eukaryotes. Nucleoid is present in prokaryotes. Right? Means, so these nucleic acids are seen both in prokaryotic cells as well as eukaryotic cells. Even this means the plasmids. Right? And these are present inside the mitochondria as well as chloroplast. Such kind of cell organelles, this DNA is present. Right? DNA is also present inside. Means DNA, RNA, they are present inside the viruses as the genetical material, as even the non-genetical material. Even it is present in viruses, non-cellular structures. They are, even though they are non-cellular in nature, they contain what? Genetical material, that is DNA or RNA. If DNA is the genetical material of these viruses, DNA viruses, they are called as. If RNA is present, they are called as retroviruses. Sometimes one group of RNA viruses are called as retroviruses. Why they are called as retroviruses is because they contain one of the enzymes, right? So that is that enzyme which can turn DNA to RNA, right? So like this, so such kind of viruses group is also containing this genetical material. Occurrence of these DNA as well as RNA. DNA is usually double-stranded one, but sometimes it is a single-stranded one, right? But RNA is always a single-stranded one, right? Such kind of DNA as well as RNAs are present both in prokaryotic as well as eukaryotic organisms, even the cell organelles, etc. Right? So this is about the occurrence of these nucleic acids. Then, what these nucleic acids are basically chemically made up of. The chemical composition of these nucleic acids is also very important to know here. Right? In NCERT, he has explained very complicatedly, so the same things in NCERT book. But so we have to make it a dilute so that you can understand it very clearly. Right? So all these historical aspects, even the definition, all these are not given in NCERT book. So he says directly they are the heterocyclic polymers of nucleotides. He gives the definition for this one. What do you mean by that one? You don't understand very easily. For that reason, there is a need of understanding the aspects, right? So very easily regarding these nucleic acids, I am going to explain all these things. Thoroughly you read all these things so you can understand the things what are given in NCERT very easily, right? So now let us start with the chemical composition of these nucleic acids now. Chemical composition of these nucleic acids. See here, chemically these nucleic nucleic acids are made up of? First one is pentose sugar. You know it better. Pentose sugar. One. And even it is made up of. Second one is nitrogen bases. And third one is phosphate group or phosphoric acid. These are made up of. These three things together form what? One monomer of this polymer, that is nucleic acids. I will explain that one, which are called as monomers of these nucleic acids and what are the polymers of these nucleic acids. So let us see them now. Pentose sugar. First one I shall go through. The pentose sugar. See here, the pentose sugar present inside the DNA is. DNA is one type of nucleic acid which contains the pentose sugar, that is deoxyribose is the name of the pentose sugar present in DNA. And in RNA, so there the ribose is the pentose sugar present in. See here, in pentose sugar, what happens? Already we have gone through the structure of this pentose sugar. See here, there we come across. So there are 4 carbon atoms are present in a ring, right? And 5th carbon is forming the CH2OH group. So this is the 5th carbon atom. Since it is pentose sugar, this is the first carbon atom here. This is second carbon atom. This is third carbon atom. This is fourth carbon atom. This is 5th carbon atom, right? So see here, OH. So this is a deoxyribose, what I am drawing here. So this is OH, here H, here H, OH, and here H. Simply, whenever you observe this one, why it is called as deoxyribose? Ribose is the name here, but deoxy is given. Deoxy means absence of one oxygen atom in the second place of. Here OH is not here. This O is omitted here. So that's why it is called as deoxyribose. Simply if you wrote this OH over here, then automatically it becomes what? Ribose sugar itself. That is the only difference over here. Ribose sugar. Its structural formula is also almost similar, right? So that's why this is the only difference between deoxyribose as well as ribose sugar. And this ribose sugar is also helpful in synthesis of ATP over here, NAD over here, NADP over here, FMN here, right? So all these are the coenzymes which are synthesized with the help of the ribose itself, right? Deoxyribose as well as the ribose sugar. On the second place of the deoxyribose molecule, one O is removed. That's why it is said as deoxyribose sugar, right? So this is one fact to be remembered. Means chemically, so these nucleic acids are made up of one pentose sugar. So what is the pentose sugar in DNA? Deoxyribose. What is the pentose sugar in RNA? Ribose is the pentose sugar. And phosphate group or phosphoric acid. See here, this name acid itself indicates that these nucleic acids, these are called as, why these are acidic is because of this presence of phosphoric acid within themselves, right? So and this phosphate group is usually present on the third carbon atom of the pentose sugar, and even it is the 5th carbon atom of the pentose sugar. It is present in the 5th carbon atom of the pentose sugar as well as 3rd carbon atom of the pentose sugar over here. So this is the fact we remember. Okay, I will show that one structurally later, right? So totally, so what it is made up of? One phosphoric acid and one pentose sugar and nitrogen bases. Even it is made up of nitrogen bases. These are nitrogenous bases which contain the nitrogen within themselves. So these nitrogen bases are of two types. One is called as purines, another one is called as pyrimidines. One is purine, another one is pyrimidine. So purines are of two types. One is adenine and guanine. Adenine as well as guanine are said to be as the purines over here, and pyrimidines are also there. Pyrimidines are of two types. What are those two types? One is cytosine, and so there are three types of pyrimidines that we come across. One is cytosine, and cytosine is there, thymine is there, and uracil is there. Uracil is there. There are three types of pyrimidines that we come across commonly both in RNA as well as DNA. See here, first of all, one more difference between DNA as well as RNA is, see here, cytosine as well as thymine are the pyrimidines present in DNA. Okay, and this thymine is replaced by this uracil in case of RNA. Means RNA consisting of the adenine, guanine as the purines, cytosine as well as uracil are the pyrimidines in RNA. This major difference between these two, you must remember this one, right? So means RNA has what? Purines as well as pyrimidines. What are the purines present over here? Adenine as well as guanine. What are the pyrimidines present over here? Cytosine as well as uracil. Here thymine is not present over here, right? So this is the difference between DNA as well as RNA. But in case of DNA, purines are commonly present are adenine as well as guanine, and pyrimidines are cytosine as well as instead of uracil, there you come across thymine, right? So these are the purines and pyrimidines of DNA. These are the purines as well as pyrimidines of RNA. This fact we known to you, right? So you must remember these aspects over here. Now we are able to understand. So what? Nitrogen bases are also present inside the nucleic acids. These are also the major components of nucleic acids, right? So this is about the nitrogen bases as well as phosphoric acid. Along with that one, what? Pentose sugar. All these together form what? So these form the nucleic acids. So but basically what these three together form, let us see now itself. Yes, now we shall start with the nucleoside. Nucleotides and polynucleotide chain. Polynucleotide chain, right? What are these nucleosides? What are these nucleotides? And what are these polynucleotide chains? So if you gone through all these one, then which are monomers, which are polymers, everything you can understand very easily, right? So let us go through this one. First one, what are these nucleosides? Nucleosides. What these nucleosides are? These are the combination of one pentose sugar and one nitrogen base together form one nucleoside. Let us see how many number of nitrogen bases are there. 4 are there totally. 2 are purines and another 2 are pyrimidines. Let us go through all those one. What is the pentose sugar present in DNA? So nucleosides of DNA we see first. Nucleosides of DNA are different and the nucleosides of RNA are different. So both of themselves we will discuss now. Right? So now we shall start with the DNA nucleosides. First one. So what is the pentose sugar present in DNA? That is deoxyribose. Oxyribose is the pentose sugar. It's a common pentose sugar present in DNA. And one nitrogen base. Which is the first nitrogen base? That is adenine. Adenine you write here. Totally these two combined together form one nucleoside. See here, deoxyribose plus adenine, so that is deoxyadenosine. Is the name of what? Deoxyadenosine is the name of the nucleoside of one DNA itself. One nucleoside of DNA. Deoxyribose is the common factor. Here guanine is another nitrogen base. Deoxyganosine is another one. Third one, deoxyribose plus cytosine forming deoxycytidine. And fourth one, deoxyribose plus thymine, deoxythymidine is the fourth nucleoside of DNA. So like these 4 nucleosides that we come across in case of DNA. Very simple one, right? So these are the nucleosides of DNA. Okay, and let us go through the nucleotides of DNA. What? Nucleotides of DNA we shall write here. So first of all we will write nucleosides of DNA and second one nucleotides of DNA. And later nucleosides of RNA, later nucleotides of RNA. Okay, so we'll finish this way. Nucleotides of DNA. See here, this is the combination of one nucleoside plus one phosphate group. So when these two are combined together, then it forms one nucleotide. What it forms? Nucleotide. See here. So first one we shall write one nucleoside. What is that first nucleoside? Deoxyadenosine. Right here. Deoxyadenosine is the first nucleoside. So add to this one phosphate group. So that what happened here itself? Deoxyadenosine monophosphate. What it became? Deoxyadenosine monophosphate. Simply you write it deoxyadenosine monophosphate. DAMP is the shortcut name of this one, first one. And second one, deoxyganosine. Deoxyganosine plus phosphate group forming deoxyganosine monophosphate. Deoxyganosine monophosphate is the shortcut name I am going to write over here. Okay, and the next one, third one, deoxycytidine plus phosphate group, deoxycytidine monophosphate, DCMP. And fourth one, deoxythymidine plus phosphate group forming deoxythymidine monophosphate. How many nucleotides are there in DNA? 4 nucleotides are there in DNA. So one is deoxyadenosine monophosphate, DAMP, DGMP, DCMP, DTMP. So these are the 4 nucleotides of DNA. Now it is very clear to us that, so what are the nucleosides? What are nucleotides? So what are the nucleosides of DNA? What are the nucleotides of DNA? So you must remember all these aspects first. So such kind of many number of nucleotides, so this DNA is made up of. That's why so many types of, 4 types of nucleotides it is made up of. Hence it is said to be as what? Heteropolymer of. So heteropolymer of what? Nucleotides. These are, so DNA so is called as heteropolymer of nucleotides. Nucleic acids are simply said to be as heteropolymers of, so what? Nucleotides, right? So these are commonly known as heteropolymers of nucleotides. Many nucleotides it is made up of. So heteropolymers, polymers are nothing but maximum number of nucleotides joined together forming a polymer. Right? So this is the fact to be remembered. Let us go through the nucleosides and the nucleotides of RNA itself, and afterwards polynucleotide check we'll see. Yes, nucleosides of what? RNA. Nucleosides of RNA. What are nucleosides? Nucleosides are the combination of one nitrogen base plus one pentose sugar. What is the pentose sugar present in RNA? Here ribose. First one I shall write here. Ribose plus one nitrogen base, that is adenine to form what? Adenosine is one nucleoside of what? RNA. Second one is ribose plus guanosine. Third one, ribose plus cytosine, cytidine. Fourth one, ribose plus, here in RNA uracil is present, then uridine. So these are the 4 what? These are the 4 nucleosides of what? So the RNA. And nucleotides nucleotides of RNA. So what are nucleotides? Here these are the combination of one nucleoside plus one phosphate group, right? Or simply you may write here one pentose sugar plus one nitrogen base plus one phosphate group. All these together form nucleotides. You may write like that also. Alright? So in some books this is the method which is given. In some book this is the method which is given over here. See here, nucleotides of RNA. Okay. So let us write what is the first nucleoside. Adenosine plus phosphate group. Adenylic acid it is. Guanosine plus phosphate group. Guanylic acid it is. Cytidine plus phosphate group. Cytidylic acid it is. Thymidine plus phosphate group. Thymidylic acid it is. So like this, there are 4 nucleotides are also seen in case of RNA, right? See here, the bond between these one is also very important one, right? What are the bonds present over here? See here, this is a deoxyribose plus one bond is there, almost with the combination of one, so nitrogen base, that is adenine. And this bond is glycosidic bond. It is also said to be as N-glycosidic bond. What it is called as? N-glycosidic bond is present between one pentose sugar and one nitrogen base. So as to form one nucleoside. One thing. And there is a combination of one nucleoside and phosphate group. There is another bond is there. There is another bond is also present over here, and this bond is phosphoester. Phosphoester bond is present over here in between one nucleoside and phosphate group. Right? So this is the fact we remembered over here. So these bonds are also very important to remember for a NEET examination purpose. Okay. So this is the fact what you should remember. So nucleosides and nucleotides of DNA and nucleosides as well as nucleotides of RNA. So these are the things that I have finished in this class. And what is polynucleotide chain? The name itself suggests it is made up of many nucleotides, right? Many number of types of different types of nucleotides it is made up of. Hence it is called as heteropolymer of nucleotides. These nucleic acids are commonly known as, right? So what are polynucleotide chains? Let us see them in the next video. Thank you.